Well, thanks very, very much, Jacob, for those warm remarks. I'm very happy to be here. And more importantly, I want to say I'm sure my father would be very happy to be here, and he'd approve very much of the program that was set up in his memory. And I want to thank Jacob for his help in doing that, too. He had a large part in it, having been a friend of my father and having worked through the whole process with my mother. I am actually something of a newcomer to genomics by that term. For the first half of my career, I worked mostly with biochemical methods, and it wasn't until really rather recently, by that I mean the early 1990s, that I began to be interested in a pure genomic genetic approach. And I became interested because I was interested in the question of how TNF synthesis was controlled. In effect, how is it that lipopolysaccharide can activate the production of TNF and other cytokines? How is it that lipopolysaccharide is detected? And that's a fundamental question of innate immunity. I seized upon a genetic model to work with at that time, a mouse that couldn't respond to lipopolysaccharide. This mouse, the C3H-HEJ mouse. In those days, it was rather difficult to positionally clone something. Sequencing certainly wasn't what it is today. One had to elaborately map a mutation by phenotype, confine it to a certain part of the genome, then go looking for candidates and ultimately track down the mutation. And that's what we did. It was a process that took about 5 years, but at the end of that time, we knew that a single amino acid change in the cytoplasmic domain of the toll-like receptor 4 would abolish the response to lipopolysaccharide, suggesting that this was the LPS receptor. And we developed other data as well, that this was the physical receptor for LPS that triggered everything that LPS could do. If we fast forward over that time, a great deal of structural biology has been done with the toll-like receptors, and we've developed drugs that interact with TLR4 in an agonistic manner. One such drug is called neoseptin 3. Interestingly, 2 molecules of the drug bind within the MD2 pocket of this receptor, which has, as a second subunit, this basket-shaped protein called MD2. Here you have 2 drug molecules binding. They always bind to each other, and they bind to critical residues within the MD2 pocket in such a way as to create a conformational change that's virtually identical to what LPS can do, though this complex of drug molecules is much smaller than LPS, and it doesn't bind to exactly the same residues. This drug works as a wonderful adjuvant, and we think of it as a candidate adjuvant for human vaccines in the future. We've developed drugs that target other TLRs in the same agonistic fashion as well, and this is a really interesting developing story in my lab. But you have to remember that it all started with a mutation and really with a phenotype. And from the year 2000 forward, I was so enamored of this whole approach of beginning with a robust phenotype and tracking down the mutation that I wanted to be proactive about it. When I moved to Scripps Research Institute in that year, I decided that we would do ENU mutagenesis and create all the phenotypes we could in every area of immunology that interested us. And we would use the old method of positional cloning to track down all of the phenotypes that seemed interesting. ENU mutagenesis in those days, as now, involved giving this mutagen to a male mouse of the black 6 strain, breeding the mouse to create a G1 that was heterozygous for point mutations induced by the mutagen. One would then cross this mouse to another black 6 female, get daughters, which could be crossed to the G1 founder itself, and then you would have G3 generation mice where there would be homozygosity for some of those mutations. We did this, and it was very difficult in the beginning still to track down the cause of phenotypes, but we made all of these interesting looking mice. We also found many mice with immune deficiencies of various types. And we learned in the process that if you see a phenotype, almost invariably it results from a change in coding sense. But this was a blind process. We didn't know how much mutation we were creating, let alone how much damage we were doing to the genome with this mutagen. My guess at the time was maybe 4 or 5 changes in coding sense per mouse, per G1 mouse. But that was all something that we had to wave our hands about quite a bit. We made small pedigrees to minimize cost in those days, thinking that we didn't need to see a phenotype more than once. Once should be sufficient for us to make a homozygous stock, propagate the mutation, and map it. During about 11 years at Scripps, we mapped and tracked down many mutations, and I go back to my old PowerPoint slides, and remarkably in those days we didn't really have a way to keep track of what we were doing other than to make drawings like this or keep Excel files. At one point in 2007, I gave a presentation remarking that we had mapped 90 phenotypes by that time over a period of 7 years. Of those 90, I said at the time, we had found the molecular defect in 69 cases, and of those, 49 were immunologic. But I have to be completely honest and say that some of these were visible phenotypes where there was a known associated immune defect of one kind or another. And to be sure, this was much slower than what we came to expect later on. Some advances began to speed things up. The biggest of these was that in 2002, the draft sequence of the mouse genome was published. And from that point forward, we didn't have to make contigs anymore. One could map, sometimes rather loosely, and all of the genes would be known from looking them up on the web. We also devised a way to sequence critical regions, exon by exon, covering only coding region. This was a program written in our laboratory to drive a robotic liquid handler in order to set up all of the amplified segments for sequencing, and then one could execute sequencing over the whole interval. And from that point, we also were able to develop software that would call the mutation. By 2009, though, it had become practical to sequence a whole mammalian genome in an ordinary laboratory. And then if one had a mouse with a clear recessive mutation that created the phenotype, the protocol was sequence that mouse and hope that there's one and only one mutation in the critical region. By 2011, it became possible to do whole exome sequencing. And that opened the doorway to sequencing the G1 mouse and knowing up front all the mutations it would transmit into a pedigree. This allowed us to eliminate obvious mutations that were well known from the very start, and that was helpful in itself. But genetic mapping for something new remained unavoidable, and this became the bottleneck of the whole procedure. The rote protocol on seeing a phenotype was, first of all, you generate a homozygous stock, then you outcross to a mouse of another strain that has molecular markers, and by that time we were using C57 black 10 for mapping because it was similar enough to black 6 that we would rarely lose phenotypes as a result of modifier effects. Then you backcross to the mutant stock. Then you genotype and phenotype the F2 animals to establish the critical region, and then you look for the mutation by sequencing a homozygote, as I described, or you might know it already if this was after the year 2011. But all of this took much too long. You can imagine that postdocs and students would frequently see some kind of aberration, and in the small pedigrees that we examined, we didn't know whether it was something genetic or non-genetic, and they would struggle sometimes for a year or two to try to see the phenotype again, sometimes successfully, sometimes not. Many more phenotypes were declared than could actually be found, and so the number of stocks we were dealing with grew in the vivarium, and we needed a new approach to automatically map the mutations much faster. By this time, it had occurred to me that if we were really confident that we were detecting all or nearly all of the mutations induced within coding region by ENU, those mutations themselves could serve as markers, and we didn't need to go through this step of outcrossing to another strain. One could, in principle, just look at statistical association in recessive or dominant models of inheritance and then declare, based on a finding of a departure from the null hypothesis of non-association, that one had found the causative mutation. So this led to a very different approach in our laboratory. Now it's the case that if we see a phenotype, its genetic cause is known at the very same time. So how do we do this precisely? We mutagenize, as we always did with ENU. We generate G1 animals, but all G1 animals these days are whole exomes sequenced in order to know all the mutations that they might transmit into the pedigree. There is no longer any guesswork about how many coding changes exist in a pedigree. Because we've done so many of them now, we stopped keeping track with a histogram in January of 2017, but the mean number is 60. And remember, with very few exceptions, if you see a phenotype or detect a phenotype, it's caused by a change in coding sense. If there are more than 30 mutations in a G1 animal, we deem that that mouse is worthy of being bred and of being studied phenotypically, and so we order an AmpliSeq panel to amplify all of the mutation sites in all the descendants of that mouse, and meanwhile we breed it to make a large number of G2 daughters and a larger number of G3 progeny. Typically 10 or 12 G2 daughters are made and systematically backcrossed to the G1. We aim to get about 50 G3 mice, and typically we reach about 43 in our average pedigree these days, which is ample for mapping. Now, all of the G2 and G3 mice are genotyped up front to know which of the mutations in the G1 sire are present and what their zygosity is. And only at that time, after genotyping is accomplished, are these animals released for phenotypic screening. While the G1 is sequenced using Illumina technology, we typically use ion torrent technology to sequence the G2 and G3 mice. This is more convenient than using the Illumina machine because one can take an AmpliSeq panel, usually for 3 different pedigrees, on one chip and determine the genotype of all of the mice in that set within about 8 hours. Now, here I've displayed, as we can do with the computer, the genotypes of mice from a particular pedigree. Across the top are each column a different mouse, and then over at the side here you see gene symbols, and coded in yellow are homozygous reference allele, in green heterozygotes, in blue homozygous mice, and by mousing over in the real program you can see the number of reference and variant calls for every mouse at every site. Here would be something typical. You can see the mouse here has 514 variant calls, 0 reference calls, and so you know it's a homozygous variant. This would be a homozygous reference allele. This would be a heterozygote. If you plot the percentage of variant calls in each mutation site in each mouse, you come up with 3 populations: homozygous variants, heterozygotes that are around 50%, and reference allele mice that are around 0%. And there's almost nothing in between these peaks. So this is a very robust means of genotyping, and it's the least expensive that we can use. An average of 88 different assays are applied to every mouse that's put into the screening pipeline. We look first for neurobehavioral problems, dystonias and ataxias, by putting mice on a rotarod. Then we test innate immune competence, then adaptive immune competence, and we do flow cytometry on the blood of every mouse. We look at the ability of NK cells and CTLs to kill based on a model target antigen. We do a glucose tolerance test on the mice too, and have found novel forms of diabetes, one of which I talked about 2 years ago. We look at allergies, at bone density and the length and morphology of bones, blood pressure, heart rate. We injure the epithelium of the gastrointestinal tract with dextran sodium sulfate using a very low dose, and we find exceptional animals that can't reestablish homeostasis in the GI tract. Then the mice are split into several pipelines. We look at their resistance to melanoma, at addiction-related behavior, at the function of the eyes, also at circadian rhythms and innate fear. And separately, pedigrees are generated for a screen that's related to autism in the mouse. Again, an average of 88 assays for every mouse. As of last Friday, while I was on my way here, I tabulated the statistics of how much mutation we had done and subjected to screening. And 138,194 coding splicing variants were fed into the pipeline for phenotyping. Those fell into more than 20,000 genes, and the total gene number in the mouse is 24,977 if you take the MGI numbers. So we've at least touched 80.4% of all genes in the genome. These mutations resided within a total of 77,310 G3 mice from nearly 2,500 pedigrees. And a total of 11,900,000 statistical tests of association between phenotype and genotype were carried out. That's roughly equal to the number of mutations times the number of screening assays to which they were subjected, and that's where we get the figure of 88. The big question that loomed over us for a long time was, how much have we really damaged the genome with all these mutations? Some mutations, of course, are functionally neutral, and we didn't know for the longest time how many of those really were functionally neutral, and we didn't have a very good way to assess the matter. For some time, we would again do a lot of hand waving, and we would look at saturation as follows. We would say we want to consider saturation in a robust sense. If a mutation has occurred, we want to have looked at it 3 times in the homozygous state, and we could model all of the mutations that we had induced in that way. And here would be the numbers of mutations over time. The red bar would be where we are at the moment, and these would all be simulation of mutagenesis curves. The green curve would be all mutations that we created that changed or could change coding sense. The blue curve would be mutations that were putative null alleles, premature stop codons, or critical splicing errors. The brownish curve would be either putative null or called probably damaging by polyphen 2. And we knew that polyphen 2 would overcall damage. Most things called by polyphen 2 probably damaging really are not damaging as far as we can discern. So we knew this was the upper limit of what we had accomplished in terms of creating phenotype. And we knew that the lower curve was probably the most conservative estimate, and the true estimate had to be somewhere between these 2 curves. So we took the midpoint and said probably we're about here in saturation. But this didn't have a very good rational basis. As you might expect, if we compare these simulated curves to the actual results of mutagenesis, and here we're looking at the percent saturation of the genome on the left, we found that a good deal of mutation never made it to homozygosity. And this is, of course, because some of the mutations have lethal effects. And the departure from the prediction was particularly extreme in the case of the curve for putative null alleles. We then decided to estimate the number of essential genes by imposing in the computations a mouse killer routine, in which we would say at random 10% of genes, we would say they have a lethal quality when mutated to a null state, or 20% or 30%, and gradually the curves would approach the actual data until they became superimposed. This led to the estimate that 34% of genes are essential for survival of the mouse to weaning age. And this comports quite well with estimates from the Knockout Mouse Project, where 32% is said to be the magic number. Now, knowing that you have such a large phenotypic genomic footprint of the lethal phenotype, that it's 33, 34% of genes, suggests that you can use lethality to estimate the likelihood that a given gene has a lethal mutation, that a given mutation really damages the protein. So this was done by taking isolated mutations that fell within a curated set of essential genes and asking for each range of polyphen scores, what is the true probability that the protein is really damaged, as manifested by a lethal outcome? The answer is that if polyphen 2 calls a mutation probably benign, there's a 4.5% chance that it actually will damage the protein based on a lethal outcome. If it says possibly damaging, there's a 9.8% chance. If it says probably damaging, there's a 17% chance that it really does damage the protein enough to create a lethal effect, if it's in one of those genes that's essential for life. Interestingly, our call of putative null or probably null, based on what I just told you, premature stops or critical splice junction errors, gives only about a 61% chance of causing damage to the protein sufficient to create lethality. And that is somewhat surprising, but it tells you that there are workarounds. One can have alternative splicing. The mutation can truncate the protein, and there's preservation of function, and so forth. When all is said and done, 17%, or almost exactly 1 in 6 ENU-induced mutations, will wreck the protein enough that it will create a lethal effect. That's a little bit less than I would have thought, but that's the fact, and it's something that we have to keep in mind as we go forward with mutagenesis. Knowing all this, one can look across all of the set of mutations that we've created, estimate all of the effects using polyphen 2 or a collection of such programs, and one can assess the likelihood that any given gene has been damaged. One can also tell how much damage has been done across the genome in any phenotype that's been studied, and therefore we know with fair precision how far we are from saturation. When we do that, and we look at mutations transmitted to homozygosity once, twice, or 3 times, we can estimate how much damage there has been done to the genome, and taking twice transmission to homozygosity as an adequate standard of testing, we can say that we've looked at 34% of all the genes in the mouse after having damaged them quite severely. Could we introduce more mutations? We certainly could. At one time, we would cross G0 males to G1 females that already carried mutations, and that would boost the number of mutations in the resulting G1 by 50%. It also had the advantage of mutagenizing the X chromosome, and that we don't do at present, and it contains 7% of all the genes. But in general, this is not a good strategy except for the ability to find mutations on the X chromosome. When you have 100 mutations per pedigree, rather than 65 or so, then lethal effects destroy a lot of the G3 generation, and that interferes with your ability to map. Remember, 1/6 of ENU-induced mutations damage the proteins, and about 1/3 of genes are essential genes for survival to weaning age. So about 1 out of 18 ENU-induced mutations is homozygous lethal. About 1/8 of ENU-induced mutations are brought to homozygosity in every G3 mouse based on the way we set up our pedigrees. And that would mean that you lose 52% of the G3 pups in utero, or at least pre-weaning, as a result of lethal effects. And we no longer make pedigrees that way. Now, I've told you we know immediately what the phenotype is caused by when we see a phenotype. And to achieve that, we needed to write software that you see here, a program called Linkage Explorer that lets you browse your entire population of ENU-induced mutations. One can select mutations based on gene, or more likely a phenotypic screen. One can look at a particular collection of mice, restrict to certain types of mutation, restrict by the number of mice in the pedigree and the number of times a mutation was seen in the homozygous state. Most importantly, you set the p-value for the association between phenotype and genotype. To take an example, we might look at a screen of flow cytometry, look at the number of CD44 positive CD8 cells, or the MFI of CD44 in CD8 T cells, and we might set the stringencies as follows. We only accept pedigrees with 30 mice or more. We only accept those cases where we've seen 3 examples of homozygosity, and the p-value is 0.0005, and then we click return, and we get a list of genes that satisfy those criteria. Here you see the trivial name that we give to a phenotype, which is always done by the postdocs. Then you see the symbol of the gene, the number of times that you've hit the gene, and the number of pedigrees in which it's been hit, the prediction about the function of the functional effect of the mutation. These estimates of excellent or not good candidate or otherwise are made by a program I'll come to shortly. And you see in these columns the p-values for an additive model of inheritance, recessive model, or dominant model. And we could click on the recessive model in this case. And that brings us immediately to a Manhattan plot. And in this case, we've hit the DIAP1 gene twice in the same gene. And if I were to mouse over in the real program, you'd see that this mutation was null, this one was not null. I don't remember what it was. But if you click on the first of the 2 mutations, you learn quite a bit about the protein. You have a graphical display of where the mutation falls and what it is. You can also look at a gene model and see that the mutation falls in a particular exon, and if there is an effect on splicing, it tells you what that is. If you want to see the data, you can right-click on the Manhattan plot, and you find that the MFI for CD8 is much elevated as compared in homozygotes as compared with heterozygotes or reference allele mice. And so you are left in no doubt that this is a causative mutation, although even at this point, if this were something really new, we would further recreate the mutation or knock out the gene in order to have absolute certainty about it. Sometimes you have higher confidence from seeing many mutation effects. The computer recognizes when a gene is hit multiple times and generates super pedigrees when that occurs. If a different allele is produced, then a gene-based super pedigree is made. If the very same nucleotide change is observed, then that's called a position-based super pedigree. This can happen usually not by chance, but most commonly because an ENU-induced mutation is transmitted into multiple G1 mice, or because there's a background mutation in the JAK stock. We've identified more than 2,000 such mutations over time, and usually you find them and then they drift out of the stock as new frozen stocks are reanimated by JAKs. Eventually, all genes will be parts of super pedigrees. At present, we're at 80 percent of genes that have been touched once, 74.2% have been touched greater than twice or more. And with multiple alleles, your confidence in the strength of the association between genotype and phenotype grows. Here would be an example of a position-specific superpedigree where, in this particular case, we saw an allele that we called skywarp in 22 different pedigrees that didn't share G0 ancestors. This was estimated to be a background mutation in the JAK stock, and the computer also recognizes that and informs you of it. So you see here that one mutation site, amid hundreds of other mutations, it gets a terrific score in terms of p-value, the likelihood that this would happen by chance alone. And if you look at the number of CD8 cells in this case, you find that homozygotes from many different pedigrees have a diminished number of CD8 cells. You see that heterozygotes and reference allele homozygotes are the same as B6 in range and distribution. Now, I would like to point out that this phenotype would have been very difficult to positionally clone the old way because there's so much overlap between heterozygotes and homozygotes for the mutation. But in this case, it simply falls into your hand, and you know that these are certainly that this gene is important for CD8 numbers in the blood. To this point, then, we've damaged or destroyed more than 34% of all the genes in the mouse, and we've tested the mutant alleles in that case twice or more in the many screens I showed you. We've named a total of 1,200 phenotypically significant alleles of 846 genes in these areas that interest us, and many are new and many are associated with human diseases. 711 mutations that fall into 513 genes affect immunity. And here I'm excluding the DSS screen, some of which are immune in etiology and some of which are not. And I just didn't have time to sort out which fall into which category, and many are simply not known yet. Of the 513 genes, 82 have been validated as causative by CRISPR-Cas9 targeting, and an additional 167 are pending. Now, you have to understand that we don't try to confirm things that are well known and obvious, and we therefore can say that something like half of the immune candidates are thought to be sufficiently novel to validate. There is a temptation when you do this kind of genetic work to let bias creep in. The reason being that you would like something that's marginal in significance to really be true. To take one example, there was a time when we were first working with this that we saw a lot of olfactory receptor mutations. And I thought, wouldn't that be interesting if chemisensors expressed on leukocytes are important in detecting microbes and they guide the processes of the immune system just as they allow us to smell. And for this reason, I tried to verify by CRISPR-Cas9 targeting probably half a dozen to a dozen olfactory receptor mutations, with the result that none of them validated, and it choked our CRISPR-Cas9 pipeline, and it was a great nuisance to us. So we had to be more objective, I felt, in what we chose. At this point, 309 CRISPR-Cas9 jobs targeting 240 genes have been completed successfully and gone through either verification or exclusion of the candidate. This gives us the basis to train a machine learning program that evaluates many characteristics of every mutation. How many times was it seen in the homozygous state? What was the p-value? What was the range of variance in the homozygotes? How large was the pedigree? What was the assay? Who was the operator who did the assay? All of these things are taken into account and considered in training the machine. One can look then, and it's best done to look using the combination of mutation and phenotype. In other words, one mutation may give many different phenotypes. And in this case, with a certain default parameter setting, where we use p.002 as the p-value and fairly strict exclusion criteria for goodness of mutations, we've verified 149 and excluded 179 mutations. And let me blow this up for you a little bit here. With these parameters, if a mutation is ranked as excellent as a candidate for causing the mutation, there's a nearly 97% chance that it will validate with CRISPR-Cas9. But at that setting, we have only about 60% recall, so we are definitely excluding quite a few mutations that would turn out to be true. With good or better, we're at 90% precision and nearly 90% recall, so we tend to submit for CRISPR targeting only things that are good or excellent. So with this tool, we've been able, at this point, looking at innate immunity, to find 85 mutations in 39 genes that were detected because of TLR signaling defects. And 16 of these genes were new. Other mutations were informative, even if not new, but those shown in blue were truly new candidates, most of which we understand mechanistically now, a few of which we're still working on. In the area of adaptive immunity, we have found more than a dozen new, very severe immunodeficiencies that I think no one in the human world would have heard of. We have here strawberry, unbalanced, skywarp, and shaman, and I'm looking at different assays here, B to T ratio in some cases, CD4 to CD8 ratio in others. But all of these have quite severe immune deficiencies, and all of them will die if infected with mouse cytomegalovirus, for example, which depends on NK cell activity for clearance. Strawberry is a multi-spanning plasma membrane protein. It has no previously known function, and in fact there are only 4 papers published on this, but none of them really get it right, and no one really noticed the immune function. Interestingly, the knockout is fully viable in this case, where it's not in any of the other 3. And basically, lymphocytes die. They undergo apoptosis when they're activated. The unbalanced mutation and the skywarp mutation, which I showed you earlier, are both defects of splicing components. In this case, the protein involved is also involved in nonsense-mediated decay, and in both of those mutants, you have an abundance of misspliced transcripts in lymphoid cells. Why the mutations are so selective for immune function when you have a viable hypomorph, as in each of these cases, we're not really clear, but that certainly has a message that we'll be looking into. The shaman mutation affects a small GTP binding protein about which essentially nothing is known. The point of all of these last 3 is that the knockouts are lethal. We produce viable hypomorphs, and we then see immune effects. And this is something that we see over and over again. It is a fact that a lot of phenotype comes out of essential genes, probably much more than out of non-essential genes, which isn't something that's inherently obvious. It might be thought that there are very few target nucleotides that could give you viability and yet create a phenotypic effect. There are other proteins involved in immune responses. This one selectively affects the T-independent B-cell response. This one gives you an elevated number of CD8 T cells and also speeds the regeneration of hematopoietic cells in the mutant as compared with the wild type after irradiation. We have an allergy screen, as I mentioned. We find some mutations like this one in a protein kinase that creates a hyper-IgE syndrome and creates hyper-IgE responses to an antigen. This one essentially eliminates IgE and does so selectively. And all of those mutations that affect DSS sensitivity have been mapped quite carefully. Many have been knocked out. And some of these affect the hematopoietic compartment, some the epithelial compartment, some both. So, as you can see, we're on our way to destroying all the genes in the genome, and this brings up the question, is this really still forward genetics, or is it some kind of hybrid? I would argue it is still forward genetics, not reverse genetics, because we still come with no hypothesis, but it's much empowered compared to the former situation when we didn't know how much damage we were doing or had done, and finding the phenotype has been made vastly easier. Formerly, in the old days of positional cloning, only one mutation was ever examined, the mutation that you found, and you didn't know what other mutations you had produced. Now, no observations related to phenotypes are ignored or forgotten, and both causative and non-causative mutations are registered, and the memory of all the mutations is kept in perpetuity. I would argue that this whole approach is rather superior to the COMP-UCOM approach of targeting genes one after another, up to a point, because most certainly in 4 years, being able in one lab to have knocked out about 33–34% of all genes and tested phenotypic effects over a broad range is much faster than could be done by the COMP program. On the other hand, we do hit a wall with small genes, and many years will go by before we truly reach saturation, because genes that are very small, 200, 300 nucleotides, are not hit or inactivated very easily. I've told you then about what we do in terms of making mice, sequencing mice, screening them. Then we have this nice informatic way of finding the causative mutations very reliably. We also have a CRISPR-Cas9 group that's constantly targeting those things that are of interest. And here are all of those people. It's a very large lab, very cooperative lab, and certainly for a postdoc, it's a wonderful position to be in. You come and you work and you screen for a while, maybe for a year, and you're likely to have 4 or 5 phenotypes that no one else knows about, and you have quite a head start in trying to understand a new piece of the immune system. Thanks very much for your attention.